This is Dr. Jose Kudakasseril. In this video, I am going to talk about the second step in genetic engineering, namely amplification of the gene. Two techniques are used. One is gene cloning in bacteria, where we use cloning vectors, and the second technique is polymerase chain reaction. Today, I am going to talk about how we clone genes using the cloning vector pBR322. pBR322 is an artificial vector which is used for cloning of the foreign gene in E. coli. It was created by two Mexican scientists, Bolivar and Rodriguez. pBR322 has the following features. It has an origin of replication called ORI, that is where replication starts to make multiple copies of the plasmid in E. coli. The next character is the ampicillin resistance gene, or ampR, which codes for an enzyme that breaks down the antibiotic ampicillin. The next character is the tetracycline resistance gene, which codes for an enzyme that breaks down the antibiotic tetracycline. Both of these are called marker genes because they are used to identify E. coli cells which contain the foreign gene. pBR322 has 40 cloning sites or restriction sites. These are places where different restriction endonucleases cut the plasmid and we can insert the foreign gene in one of those sites. The other character is the rope, which is the full form is repressor of primer. That rope determines how many copies of pBR322 are produced in E. coli. The usual number is 20 copies. If you modify rope, you can increase the number of copies. The actual picture of pBR322 shows all the 40 different cloning sites. These sites are also called restriction sites. Now, in pBR322, we usually insert the foreign gene inside the tetracycline resistance gene. BAMH1 is a restriction endonuclease that cuts inside this gene. BAMH1 is obtained from the bacterium Bacillus amyloliquefaciens. When the gene is inserted inside the tetracycline resistance gene, the tetracycline resistance gene becomes inactive. It does not produce the enzyme anymore that breaks down tetracycline. This is called insertional inactivation. In the procedure, we take along with the buffer pBR322 restriction endonuclease that cuts inside the tetracycline resistance gene, the foreign gene, and DNA ligase. Here, what happens is that some of the pBR322 take up the foreign gene, others will not. So we get two types of pBR322, those with the foreign gene and those without the gene. Then these pBR322 are incubated with E. coli. We give E. coli a heat shock treatment in which we heat E. coli to 42 degrees centigrade and suddenly cool it to 0 degrees centigrade. We also give a calcium chloride treatment. These treatments increase the permeability of the E. coli membrane so that pBR322 can go inside E. coli. Some of the E. coli will take up pBR322, others will not. So we get three types of E. coli. First one is called the non-transformant, which has not taken up the pBR322. The other two are transformants. But the first transformant is called the non-recombinant because it has only pBR322 in it. It does not have the foreign gene. The third one is the recombinant bacteria, which contain pBR322 and the foreign gene. We are interested only in the recombinant bacteria. So let us see how we select out the transformant, or we can say recombinants, from the rest of the bacteria. E. coli cells are cultured on potato dextrose agar medium, which is the usual medium used to culture bacteria, containing the antibiotic ampicillin. The E. coli cells divide and form colonies. Each colony will have thousands of cells in it, all derived from one E. coli. The colonies are white in color and they project from the medium like this. Just remember, each colony will have only one type of E. coli in it. Because of the presence of ampicillin, non-transformant cells will die on that medium because it does not have pBR322 in it. pBR322 has the ampicillin resistance gene. Because of the absence of that gene, the non-transformants will die on that medium. This petri dish is called the master plate. Now, the transformants which are growing on the medium are transferred to another medium containing tetracycline. The technique is called replica plating. In replica plating, we take a wooden disc, attach it to a handle, cover the wooden disc with velvet cloth, and it is sterilized in an autoclave to kill all the bacteria on the velvet cloth. It is then put on the top of the petri dish so that the velvet cloth touches all the colonies. Remember, all the colonies are projecting upwards. So few cells from each colony will attach to the velvet cloth. This device is then put on another medium containing the antibiotic tetracycline. On the tetracycline containing medium, recombinants will die because when the foreign gene is inserted inside the tetracycline resistance gene, that gene becomes inactive and it does not produce the enzyme that can break down tetracycline. So they will die on that medium. Those which survive on that medium are the non-recombinants. This petri dish is called the replica plate. Now we compare the master plate and the replica plate. As shown in the diagram, two colonies shown in yellow color found in the master plate are missing in the replica plate. Those are the colonies we are interested in. They are selected from the master plate. Then with a spatula, we take out the colonies and culture them so that when E. coli multiplies, pBR322 multiplies and the foreign gene multiplies. So we get large quantities of the foreign gene. This technique is called gene cloning in bacteria. Thank you for listening to my video.
I have published some books for students appearing for the NEET exam. One is Handbook of Biology, Volume 1, which describes all the chapters in class 11, and Volume 2, which describes all the chapters in class 12. And also I have published a book, Biology in a Nutshell, which includes all the chapters. These books are available on Amazon. I am giving the link below.